Hello, my name is Ciprian Crainiceanu. I'm a professor of biostatistics at Johns Hopkins University, and I would like to thank the organizers of the symposium for inviting me to give this talk. Today, I'll talk about dynamic prediction of voxel intensities in multiple sclerosis. The general idea is that lesions develop in the brain of individuals who have multiple sclerosis, and these processes that are associated with a dynamic change in the lesions, in their occurrence and change in the lesions, may be associated with treatment or immune responses. We are particularly lucky to have a very interesting data set. The data set was originally collected at NINDS by Danny Reich and his team, and was processed, including image segmentation and other components by Elizabeth Sweeney. The data consists of 48 study participants who had a total of 751 lesions that occurred over the course of the study. These lesions had more than 180,000 individual voxels or locations in the brain, with more than with around 28 number of visits per person and approximately 30 days between scans. This is an absolutely exceptional study with a lot of data at an individual level, so that we can look dynamically at what happens at an individual over time. So just to get a better idea about what happens, I decided to take one individual person and show what happens from an intuition perspective in terms of the data collection. So this is the general outline of a brain, and the 5 locations indicated by lesions from 1 to 6 are from 1 to 5 are the lesions that occur at some point during the study. So this is day 0 from baseline. So the colors here show the intensity in the baseline, and the light blue and the darker blue identify whether or not this particular location had higher or lower intensity. So this is a second visit, which is about 28 days from the baseline, and we see that the lesions haven't changed that much. The shades of blue stayed about the same. At visit 5, which happens about 70 days from baseline, lesion 1 occurs, is identified. So you see these shades of yellow and red, which are high intensity on the flare images, and identify the fact that this was a lesion that was detected 7 days from baseline. 105 days after baseline, nothing changes substantially, maybe a little bit in lesion 1. There is no other lesion that has occurred yet. Lesion 2 is detected about 200 days after baseline. So you see that lesion 2 is now has higher intensities in flare. Lesions 3, 4, and 5 have not developed yet, have not occurred yet. At the following visit, which is 385 days, about a year later, lesion 1 continues to be there, and lesion 2 actually becomes a little bit less intense than before. Lesions 3, 4, and 5 now occur and are identified at these visits. So, and this is what happens 420 days from baseline. So what is the main problem that we are interested in? Here I provide a schematic of the data science problem that we are faced with. On the x-axis in both graphs is the time from lesion identification. On the y-axis, we have what we call the normalized flare intensity of the image. I can talk more about that if you have questions about exactly how we normalized. And what I show in each plot is just one of the voxels, one location in one of the lesions in the brain. And what you can see in the left panel is that this lesion intensity is before the lesion was detected. This is in this area right here. Vary up and down, so they go up and down. They have a natural variation around the baseline. And then the moment when the lesion is identified, there is a jump in lesion intensity, and then there is a natural slow decrease over time in this particular lesion. This doesn't have to be the case, but this is how this voxel behaves in particular. And we have an area which is called recovery. And that area is basically defined by being, in this case, about 1 standard deviation away from the mean in baseline. So look at the standard deviation of these observations in baseline. And we said if the observations start to fall within 1 standard deviation of baseline, then we say that there is recovery. So this is the practical definition for that. It could be changed depending on the particular application. In the right panel, I show the same thing, but this is for the different voxel. It behaves slightly differently, has less variability in the baseline, also goes up to about 3 to 4 standard deviation from its baseline variation. Then it slowly starts to go down, but it doesn't go into this area which we define to be recovery. So this is kind of the name of the game. We have these images over time, and each image represents the intensity and the voxel, many voxels along time. And what we want to do is to predict what happens in the future to these voxels. So to translate this into specific data science components, we have that the outcome in this case is the future voxel intensity, and the prediction is what is the history of that particular voxel's intensity, as well as what happens in all the other voxels before this particular time. So we learn from the other voxels to predict what happens in this particular voxel and understand how this lesion may develop into the future. We are also interested in time to voxel recovery. So if we're at a particular time in the voxel history, we would like to know how much longer do we expect, or what is the probability for that particular voxel to recover, to get into what I showed earlier to be the green area. Again, what we have to predict that is the history of the voxel intensity and the voxel population dynamics. So this is what it looks like in terms of prediction. Again, here are two different lesions. So on the x-axis is time, again, 0 is time before lesion has occurred. Time 0 is when the lesion is first identified. So x-axis is, as I said, time, and y-axis is intensity, in this case, intensity in flare. And in gray area, in gray lines, you see each individual voxel. So there are multiple voxels shown here, not all of them, but some of them in this particular slice. And the darker black line is one particular voxel, the one that is indicated right here. So it is this voxel right here, this voxel. So in red, what we show is the predicted values of the voxel intensity given the data only up to day 90. So I only look at the data up to this time point, and this is what I predict. So some of these voxels are relatively well predicted, and some are not, though I'm happy to talk about why we actually believe that the predictions are probably more reasonable in this particular example than the observed data. So this is fundamentally how it works. You can, of course, not only predict at time 90, but you can also cut the data at any time point when you have it, and then predict from then on. The models that we use are not super complex. They use essentially a dynamic linear model approach, and they can be easily implemented in standard software. Another thing that we may be interested in is instead of looking into what is the actual recovery of the entire voxel, we are interested in to predict what is the probability of recovery beyond a certain time point t. So in this case, again, we have the lesion incidence happen here. This is the history of the data that we have, and what we do, we predict that the recovery time, the time it takes for the voxel intensity to get to that level that I showed earlier, to be greater than time t. So this is a little bit more complex in terms of what it is that we're measuring, but for those who are familiar with survival data analysis, it is just a way of writing this approach. What is different here is that our data, what is different here from standard biostatistical methods for prediction when you have a biomarker and time to event, the difference is that in this situation, we actually have observations that are quite dense. We have a lot of measures. And I do believe that in the future, in neuroscience, we will have more and more observations, sometimes even daily. But, you know, it really depends on the application. But in the future, we'll have more observations that are happening at closer times, especially in clinical applications. So that's everything I have. Just wanted to give you an overview of the type of problems and the type of solutions that we are thinking about. We do think that this particular data set is absolutely extraordinary, and we'd like to thank Danny and his team at NINDS, Danny Reich and his team at NINDS for doing this fantastic job of putting things together. Also to Elizabeth Sweeney, who has been crucial in extracting this data and putting it together, organizing it, and make it usable and make it useful to us and to others in the same time. And also, I'd like to thank Jordan Jones and Andrew LaRue, my former PhD students, who wrote 2 fantastic theses on this. Thank you very much. I look forward to meeting everybody.